Another type of motility that is seen in case of bacteria is called as gliding motility. That means bacteria can move without the help of flagella, whether it is exoflagellum or endoflagellum, which we have seen in the last lecture. So, this is particularly the movement of bacteria on solid surfaces which has got some slime, means sticky substance. So, this movement of bacteria, which is very slow as compared to flagellar movement on the solid substrates, it is called as gliding movement or gliding motility. So there is a class of bacteria which generally do not possess flagella, but still they are able to move, and these bacteria are classified as gliding bacteria. So these gliding bacteria, they move with the help of movements in the cell wall with the help of fimbriae or pili. So sometimes pili or fimbriae, they may be involved in movement of bacterial cells. With the help of slime, these bacteria can move, and this is called as gliding motility. In this diagram, this is the bacterial cell. This is the solid substrate. This is solid substrate. This is in this diagram it is shown that it contains flagellum, but generally gliding bacteria, they move with the help of pili, which are the short appendages. This is slime, the sticky substance that a bacterium is producing. The mechanism of gliding is just similar to the mechanism of movement by a snail. So how that snail moves? It moves on solid substrate and it secretes slime. And using this slime, this snail, it can glide on the surface. So in the similar mechanism, this bacterium, it produces slime and it moves in this direction like this. So in the circular fashion it moves and then it moves ahead. Sometimes if bacterium possesses flagellum, so this with the help of this flagellum it can hold the surfaces and it can push itself on the solid substrate like this. So with the help of this slime and movements in the bacterial cell wall in the circular fashion, it can move ahead on the solid substrate like this. This is how it moves in the front direction on the solid substrate with the help of presence of slime. It can move from here to here. So this movement is very slow. So per minute this bacterium, it can move only up to a few micrometers. So this movement is very slow. So this movement is without flagella. Flagellar movements are very fast. So far we have discussed about the movement of bacteria with exoflagellum, movement of bacteria with endoflagellum, and this is the class of bacteria which moves in absence of flagella, and these bacteria are called as gliding bacteria. Bacteria with endoflagellum are called as spirochetes, and in general many bacteria possess exoflagella. Now we'll discuss about the structure of flagella in eubacteria. So eubacteria are the bacteria which are divided into three categories: gram positive eubacteria, gram negative eubacteria, and mycoplasma. Mycoplasma is a class of bacteria which do not possess cell wall. Gram positive bacteria and gram negative bacteria are the bacteria which possess cell wall. We'll be discussing more about the structure of cell wall later on. Now, there is a difference between the structure of flagellum in case of gram negative bacteria and gram positive bacteria. Let us study the structure of flagellum in case of gram negative bacteria. In case of gram negative bacteria, so this part, that is cell wall, it is enlarged here. The innermost component is cell membrane, and we know that this cell membrane, it is made up with phospholipid bilayer. This is phospholipid bilayer. External to that is cell wall. So in case of gram negative bacteria, we have already learned that the percentage of peptidoglycan is less. So this is the layer of peptidoglycan here. So there is presence of less percentage of peptidoglycan in gram negative bacteria. So here this is the low concentration of peptidoglycan layer. Gram negative bacteria contains one more layer external to this peptidoglycan, and that is called as outer membrane. This outer membrane is made up with this phospholipids and this component which is called as lipopolysaccharide, that is LPS. So this is the composition of cell wall in gram negative bacteria: cytoplasm, less concentration of peptidoglycan, and high concentration of lipids, high concentration of phospholipids, and high concentration of lipopolysaccharide. So understanding the structure of flagellum in gram negative bacterium, for that we need to understand the structure of cell wall of gram negative bacterium. So cell wall, it contains very thin layer of peptidoglycan and a phospholipid bilayer and presence of lipopolysaccharide. So flagellum, this flagellum, it is present in this cell wall. So let us understand the arrangement of flagellum. So this flagellum, it consists of three major components. This is the external part, this one. This is called as filament. We have already learned in the chapter of proteins, structure and function of proteins, the structure of this filament, which is made up with flagellin proteins. So these circles you can see, all these are flagellin proteins. This is called as filament, the first component of flagellum. The second component of flagellum, it is called as hook, Hook, hook. So this hook is a junction between this filament and the next component. The third component of flagellum is called as basal body. This is basal body. So this whole part, this is called as basal body. So this basal body, it consists of rings. these rings and central rod. This central rod is attached to this hook. These rings acts as bearings and which helps the movement of this filament in the circular fashion. So this is filament, hook, and the basal part is called as basal body. Basal body in case of gram negative flagellum, it consists of rings. This is the first ring which is going to sit or embedded in this outer membrane of lipopolysaccharide, and therefore this ring is called as L ring. L stands for lipopolysaccharide. So the location of this ring is here. Then, as I said, it consists of a central rod and these rings are arranged around this central rod. The second ring. This second ring is present or embedded within this peptidoglycan layer, and therefore this ring is called as P ring. Next ring is at the bottom. This ring, it sits on the cytoplasmic membrane, and therefore so it is just bound at the upper side, external side of the cytoplasmic membrane. Therefore this is called as S ring. S stands for superficial. So because it is just present on the membrane, it is called as superficial ring. And the last one is located within this membrane, and therefore this ring is called as M ring. So we can see here the structure of flagellum is it consists of three parts: filament, hook, and basal body. This basal body consists of four rings: L, P, S, and M. L, P, S, and M. So this is the structure of basal body in case of gram negative bacteria. This is the structure of flagellum in gram negative bacteria. Now let us combine this structure with this structure of flagellum so that we can understand how exactly this flagellum it fits in the cell wall of bacterium. So we'll separate this for our convenience, and then this flagellum it fits like this. So this is embedded in lipopolysaccharide, that is called as L ring. This is in peptidoglycan, so it is called as P ring. This is superficially present on the cell membrane, so it is called as S ring, and this is embedded in membrane, and that is why it is called as M. So these rings are L, P, S, and M. So we need to remember that flagellum in case of gram negative bacteria, it consists of four rings, and these are L, P, S, and M. So the whole structure of this bacterial flagellum, including the cell wall and cell membrane, it appears like this. So this is the complete structure of flagellum. We can see this is outer membrane, that is lipopolysaccharide. So L ring is there, P, that is peptidoglycan. Here the space between cytoplasmic membrane and outer membrane. This space is called as periplasm. So you can see that peptidoglycan is present in periplasm and this P ring is embedded in this peptidoglycan. Then this is superficial ring which is bound to membrane ring S and M ring. These are the accessory proteins which are present which supports these S and M rings. This is MOT protein and fila protein. So we are not going into the details of that. What you need to remember is this structure filament which is made up with flagellin, hook, then this is basal body which consists of L, P, S and M with central rod. Now the question is how exactly this flagellum it moves. So the movement of this flagellum or this filament is not like this. It is in the circular fashion. So as the arrow is shown here, so this flagellum movement is like this, and these are the rings which acts as bearing and that supports the movement of this filament. So movement is like this. So that is about the structure of flagellum in gram negative bacteria. We can further elaborate this. It can be, this is another beautiful diagram you can see here. This is filament, this is hook. You can see the movement of this hook is like this. So filament is moving like this and not like this. Okay, so this is how it moves. Then this is the basal body which consists of rings L, P, S, M. So as it is mentioned, L is present in lipopolysaccharide membrane. This is lipopolysaccharide outer membrane. P is present in peptidoglycan layer. There is peptidoglycan layer here, a thin layer, and this is present on the cytoplasmic membrane, that is S ring, and this is present within the membrane. It is called as M ring. So this is the structure of gram negative bacteria, how it appears.